This lecture will cover protein synthesis. It will cover from DNA to RNA to protein. Here's a quick overview of cellular production and the levels of organization inside a living organism. Everything starts at DNA. That's step one. DNA is transcribed and then translated, and using amino acids are converted into proteins. Proteins are come together to make organelles. Organelles come together to make cells. A group of cells come together to make tissues. Groups of tissues come together to make organs. Groups of organs make organ systems. And organ systems come together to form an organism. Shape is really important. The DNA determines the amino acids, which determines the proteins. At the basic chemical level, this will determine the shape of the protein, which will determine the function later down the levels of organization. This slide covers a pathway of production, the DNA to protein. So nucleus is stored in the DNA. The DNA stays in the nucleus. Here is a process called transcription, where DNA is transcribed to mRNA, called messenger RNA. This mRNA moves outside of the nucleus through the ER. The mRNA will stop at a ribosome. The ribosome reads the RNA and then will assemble amino acids into proteins. This process is called translation. The protein will travel into the ER to the Golgi apparatus. The Golgi packages the protein for storage or shipping. Remember, it's the UPS of the cell. Storage. The protein will stay in vessels in the cytoplasm until needed. Shipping. The vessels will attach to a cell membrane and undergo exocytosis and release it to the outside environment. Now we're going to go over the actual steps of cellular production, called protein synthesis. The first step of protein synthesis is called transcription. DNA can only exist in the nucleus, so in order to get genetic information out into the cytoplasm, DNA needs to be converted into RNA, which is another nucleic acid that's very similar but has some subtle differences that we'll explain in the next slide. The process of changing DNA into an RNA is called transcription. Now we're going to take a closer look at RNA. RNA is a nucleic acid, and it has a phosphate and sugar backbone, just like DNA. But instead of having the sugar being deoxyribose, the sugar in RNA is ribose, so ribonucleic acid. One of the nitrogen bases is different as well. Instead of having thiamine like DNA, you have uracil. But the same base pairing rules apply. A doesn't match up with T because it doesn't exist. It matches up with uracil instead. And also, RNA is only a single helix compared to the double helix of DNA. There are three major types of RNA we'll talk about. mRNA, which goes from the nucleus to the cytoplasm, where it meets up at a ribosome. tRNA, which is transfer RNA, which helps bring amino acids to the ribosome. And rRNA, which is ribosomal RNA, which is used to help make ribosomes. The process of transcription. DNA in the nucleus will unzip, creating a little bubble. rRNA polymerase, which is an enzyme that reads DNA code from the 3' to 5' direction, and will attach the appropriate RNA bases to it. Remember, it will match up in the complementary pairing. So if there's an A on the DNA, there will be a U on the RNA. If there's a C on the DNA, there will be a G on the RNA, and so forth. This will create a chain of mRNA. This mRNA will move out of the nucleus through the ER and stop at a ribosome. Step 2 in protein synthesis is translation. The mRNA is translated into amino acids, and a chain of amino acids will make a protein. This happens at the ribosome. Remember, a ribosome is made up of a bit of protein and a bit of rRNA, which is ribosomal RNA. The rRNA helps read the mRNA, and the appropriate tRNA, which is transfer RNA, will bring specific amino acids to the ribosome, where they can be combined together to form a chain that will eventually become a protein. RNA codes for amino acids in what's called codons, or triplets. They're called triplets because they are a series of 3 nitrogen bases, and those letters will code for specific amino acids. The 3 bases are called codons. The rRNA reads the mRNA and finds a corresponding tRNA. So the tRNA has what's called an anticodon. It's the opposite set of base pairs that would be on the codon, which is on the mRNA. The anticodon corresponds to the codon sequence on the mRNA, and then we'll find the corresponding amino acid to put in the chain. Using the 4 letters and putting them to combinations of 3, there are 64 possible codons. There is 1 codon that codes for start, and there are 3 stop codons. So there are 60 different codons that code for only 20 different types of amino acids. This provides a little bit of flexibility in the codons and helps prevent against genetic mutations in proteins that are produced later on. You can find more codon facts on page 237 of your textbook. So a summary of what we've learned so far. DNA replication is the process in which DNA copies itself. This is for mitosis and meiosis. DNA will unzip using DNA helicase. You have DNA to DNA pairing rules, and it'll make 2 new DNA strands. Each new DNA strand has a bit of the old DNA and a strand of the new DNA. Then we learned about transcription. This is in protein synthesis. This is when you take DNA and you make it to RNA. DNA unzips, and then you have your DNA to RNA base pairing rule. Uses mRNA when it's completed, and the mRNA moves out of the nucleus. And lastly, you have translation. This is RNA to amino acids to protein. This occurs at a ribosome, where RNA reads codons on the mRNA. The tRNA has an anticodon and the corresponding amino acid. The amino acid chain makes the protein. And that's a summary of DNA replication and protein synthesis.